Hi and welcome to my channel, Revise A-Level Biology with Christine. That's me, and I'm going to focus on OCR specification. In this video, I'm going to focus specifically at prokaryotic cells. So prokaryotic cells are unicellular, which means they're made up of one cell. And you have to be able to distinguish some key features inside a prokaryotic cell, which therefore distinguishes that what you're looking at is a prokaryote. So the first thing we're going to identify is the single circular DNA. Now, it is a single circular DNA that is, you can either say naked or not associated with histone proteins. And that's when you are describing it in comparison to a eukaryotic organism. So this single circular DNA is not held within a nucleus. It is within the cytoplasm in a very specific region called the nucleoid. Now, not only do prokaryotes have this circular DNA, they also have extra DNA in the cytoplasm, and they're in the forms of what's called a plasmid. So a plasmid is a smaller circular DNA which contains genetic material. Now, the next thing that we need to identify is one of the organelles inside the cell, which is the 70S ribosomes. Now, the 70S ribosomes are important for protein synthesis, which I'll do a whole video on how protein synthesis occurs. But you need to understand that ribosomes are the site of protein synthesis. And when we look at prokaryotes, they have 70S ribosomes. Now, the reason they have 70S ribosomes is because their ribosomes are smaller due to the fact that these cells are smaller than the eukaryotic cells. Now, the next thing we need to know is that the cell also has a plasma membrane. So the cell surface plasma membrane is the barrier which is going to ensure that the cytoplasm is kept inside the cell, and all the different organelles, all the different structures are held inside the cell plasma membrane. So the membrane actually acts as a barrier, and that barrier is a selectively permeable barrier. So we'll talk about that when we look at biological membranes. Another distinguishing feature of a prokaryotic cell is the fact that the cell wall is made up of a peptidoglycan. Now, that's basically a polymer of amino acids and sugars held together. So it's very specific in the structure of that cell wall. The cell wall maintains the cell shape, and therefore that allows for the cell to withstand certain environments. So the cell wall is permeable, and it's made up of these amino acids and sugar, and you need to know that it's called the peptidoglycan. It can also be called murin, but in the exam, if they were going to ask you, you would want to use that term peptidoglycan. Now, the next thing that we need to talk about is the presence of flagellum. So flagellum is a part of the cell which allows the cell to move around. So it's made up of protein fibers, and that allows the cell to move to different locations. It also has these extra hair-like structures which are called pili, but they are not expecting you to know that for your exam.